This is an overview of an article entitled Interactions Between Regulatory Variants in CYP7A1 Promoter and Enhancer Regions Regulate CYP7A1 Expression, published in the October 2018 issue of Circulation, Genomic and Precision Medicine. Cholesterol 7-alpha hydroxylase, or CYP7A1, is the first and rate-limiting enzyme in bile acid biosynthesis from cholesterol. It represents a principal cholesterol removal pathway in the body. CYP7A1 is tightly regulated by bile acids, lipids, and transcription factors, all critical to cholesterol catabolism and bile acid homeostasis, thereby regulating lipids, glucose, and energy metabolism. In genome-wide association studies, CYP7A1 single nucleotide polymorphisms, or SNPs, associate with total cholesterol and LDL cholesterol levels, gallstone diseases, blood deoxycholic acid levels, and other phenotypes. Causative genetic variants and underlying mechanisms remain only partially understood. Multiple studies implicate a common CYP7A1 promoter SNP, RS3808607, with the minor G allele shown to exhibit higher transcriptional activity than the major T allele in reported gene assays. However, increased promoter activity of the RS3808607G allele is inconsistent with expected phenotypic results in CYP7A1 association studies. The G allele is also associated with higher lipid levels, which would be expected for low CYP7A1 activity, not high activity. These conflicting results indicate that either RS3808607 is not the causative variant, or more than one functional variant regulates CYP7A1 expression. To identify regulatory CYP7A1 regions and their interactions, the authors employed chromatin conformation capture, or 4C, assays, followed by CRISPR-mediated genome editing. They then screened for SNPs located in regulatory regions, testing effects on hepatic CYP7A1 expression by measuring allelic messenger RNA expression imbalance. They also tested the effects of the variant alleles of the SNPs in reporter gene assays. Using 4C assays in human hepatocytes, the authors identified 3 regulatory regions downstream of the CYP7A1 gene, the second of which they were able to subdivide into 2 smaller regions, yielding a total of 4 candidate regions, specified as R1, R2A, R2B, and R3, as shown on the left. Chromatin immunoprecipitation followed by quantitative PCR, or CHIP-qPCR, confirmed interactions of the candidate regions with the P300 protein, consistent with transcriptional regulatory activity, as shown on the upper right. They then used CRISPR to delete each of the 4 regions in HepG2 human hepatoma cells, and they found effects that varied from decreased endogenous CYP7A1 expression to increased expression to a neutral effect, as shown on the lower right. This defined R2A and R2B as novel enhancers, and R3 as a novel repressor for the CYP7A1 gene in hepatic cells. For the 3 regions with enhancer or repressor activity, these regulatory effects were largely confirmed in luciferase reporter assays, as shown on the upper left. The authors then searched for regulatory SNPs located within the regions. They focused on 4 SNPs and found that only for RS9297994 did allelic variation show a differential effect in the luciferase reporter assays, with a minor G allele showing reduced expression, suggesting the SNP might be a causative variant. The authors then returned to the already known promoter SNP RS3808607, for which the minor allele confers increased expression. They tested the CYP7A1 promoter and the novel enhancer together, with different allelic combinations of RS3808607 and the new SNP, RS9297994, in luciferase reporter assays. They found that the 2 SNPs contributed equally in a combinatorial fashion to expression. The authors then confirmed the combinatorial effects of the two SNPs, the old one and the new one, in human liver samples, using hepatic CYP7A1 messenger RNA allelic expression imbalance assays, as shown here. The graph on the lower right demonstrates this most clearly. Further details about these experiments are available in the online version of the paper. Finally, the authors assessed for associations between the genotypes of the two CYP7A1 regulatory SNPs, singly or in combination, with clinical phenotypes in 3 cohorts. Shown here are the results for the CATHGEN cohort. What is clear is that when each SNP is used alone, there are no strong associations with phenotypes. In contrast, when a 2-SNP model is used, there are associations with a coronary artery disease index score reflecting the extent and anatomical distribution of disease, with myocardial infarction, with diabetes, and with hypertension. These findings confirm that both of the SNPs are causative and that the genotypes of both SNPs need to be considered in assessing the effects on CYP7A1 expression and downstream phenotypes. In conclusion, 4C assays in CRISPR-mediated genome editing in HepG2 cells revealed novel CYP7A1 enhancer and repressor regions. SNP screening with reporter gene assays and allelic expression imbalance in liver samples identified and confirmed 2 functional SNPs, one already known and one new, with opposite effects on hepatic CYP7A1 messenger RNA expression. And finally, that only a 2-SNP model, not each single SNP alone, showed associations with clinical traits. The complexity of the regulation of CYP7A1 gene expression demonstrated in this study is likely to reflect many, if not most, loci identified in genome-wide association studies.